Oh, yeah.
When you first got here.
Nice to meet you.
Yeah, I'm super excited to hear your talk. That's a run right at the end of your talk. I love the chat since.
Yeah, no, I'm in town, so
Yeah.
send me an email whenever.
Yes.
Okay, let's get started, guys. This is our first talk of the semester, so it's good to have everyone back. We are pleased to welcome Dr. Sara Miller, who received her PhD from the University of British Columbia and was a postdoctoral fellow and then research scientist at Cornell University. Sara is an evolution ecologist and just started a new lab at the University of Missouri St. Louis in August, where she'll be studying social wasp diversification. We're very excited to hear Sara talk about novel traits and the origins of diversity in social wasps.
Great, thank you. Yeah, no, I'm really excited to be here today. So kind of before I launch into my talk itself, over the last couple of years, I think both myself and others in the field have begun to appreciate kind of how the background of scientists can affect the way that science happens. And so at UMSL, we've started this tradition of the first time that someone gives a talk as in our seminar series, that they provide kind of a bit of a personal introduction about who they are and how they got to where we are today. So I'm going to continue that tradition here. So I am actually a St. Louis native. I was born and raised here. So this is 7-year-old me sporting my favorite St. Louis Zoo t-shirt. So I was a child of academic parents, but for better or worse, spent my formative years in the St. Louis public school system. So I went off to college at the University of Chicago, graduated as many undergraduates do, not exactly sure what I was going to do with my life. So I came back to St. Louis for a couple of years and worked as a technician actually at Washington University in a lab that studied neural muscle diseases. So I basically, after a couple of years of this, decided that cell molecular tract was not for me. So I went on and actually did a master's focusing on ecology and evolutionary biology at UMSL to kind of switch my career focus a little bit. So I went on and did my PhD at the University of British Columbia in Vancouver, focusing on biotic interactions in three-spine stickleback. So three-spine stickleback are sort of maybe the poster child of the species that has a large degree of ecological differentiation, but maybe not a huge amount of morphological differentiation. So I switched systems for my postdoc at Cornell to paper wasps, which I think present a really nice contrast, which I'll be able to convince you about during the course of this talk, where they have pretty subtle ecological differences, but pretty dramatic phenotypic differences. Oh, and so there's my new lab that I started is right back there. So if you're in the neighborhood, come by and say hi. Yeah, so a lot of my scientific past and future has been really focused on this question. So how is biological diversity created? So why do we have so many different phenotypes of organisms? Why are there so many species? So there are quite a number of mechanisms that explain that, but for the purposes of this talk, I'm going to focus on sort of 3. So the first mechanism by which we can create biological diversity is we can evolve new traits. So traits in particular that allow organisms to access new niches can be a source of diversity, both from the evolution of that trait, providing diversity, and also in these new niches, species are often freed from competition, which can lead to subsequent speciation and diversification. So just a couple of examples of this. So one classic example is the evolution of birds' wings, which allowed birds to access a whole suite of new sort of resources. Another example that came from a very, very recent paper that I like is a shift in wrist bone for salamanders that allowed them to move from a terrestrial to an arboreal environment. Okay, so another way that we can create biological diversity is through adaptation to our abiotic environment. So differences in environmental conditions can promote the opportunity for diverse traits to evolve. So this can happen through phenotypic plasticity. So an example that I think is pretty cool is this one, looking at developmental polyphenism in caterpillars. So essentially, to be cryptic on your background, if you do, depends on what time of the season you emerge. So sometimes you want to be kind of brown and stick colored, and later in the season, you want to be kind of green and leaf colored. We can also get adaptation to the abiotic environment that leads to local adaptation. So an example of this would be three-spine stickleback from lakes with high predation pressure versus low predation pressure. We can see the evolution of differences in armor traits. So sort of longer spines, more intense lateral plates, and those with higher predation pressure. So the last source of biological diversity I'm going to touch on today is competition from the biotic environment. So competition for resources can lead to selection for diversity. So this is essentially the way that character displacement works. So to kind of walk you through what a cartoon example of this looks like. So here we have two species that are going to be allopatric. And so in allopatric, they're not going to come into contact with each other. So they can have sort of the same traits, utilize the same resources. But when they come into contact for each other, all of a sudden this region where we get the most number of individuals with that trait value are going to be competing not only with individuals of their same species, but with the other species. And so this competition can really promote the evolution to different traits to avoid competition. So we can get this through a change in our trait means, or we can also get this through a change in our trait variances. So again, a stickleback example, because I have trouble resisting throwing a stickleback example out when you can put one in. So this is a pretty classic example of character displacement in this trait called gill rakers, which stickleback use to filter zooplankton. You can think of it kind of like baleen in a whale. So for most, lakes have a single species of stickleback, and so they have a unimodal distribution of this gill raker length. But in a few select lakes, we get two species of stickleback, so one in kind of the muddy benthic environment and one in a sort of open water limnetic environment. And now we see a bimodal distribution of this trait. So another thing I think that's important to point about character displacement. So we see it a lot in sort of ecological contexts, but we can also see character displacement for other types of resources. So this could be competition for nesting sites. This could be things that are used in mating contexts. So frog calls are another example, things like that. Okay, so for this talk today, I'm going to tell you sort of about 3 different projects touching on these different aspects of biological diversity. So the evolution of a novel trait in a single species of paper wasp. We're going to focus on the effect of the abiotic environment across North American species of paper wasps. And then we're going to zoom out and look at biotic interactions across the entire paper wasp genera. Okay, so starting with the evolution of a novel trait. Okay, so how do novel traits evolve? We actually don't know much about how to answer this question. So even basic things like, are novel traits mostly the result of the change in one key gene, or is this changes in multiple different genes? Are novel traits the result of the origin of new mutations, or is this co-option of existing variation? And when we see genes that are involved in novel traits, did they change? Was the evolution of these genes, did this occur sort of as a gradual stepwise process where we see kind of an improvement of our novel trait, or was this more of a punctuated event that happened kind of in one shorter time period? And the reason that we don't know much about the evolution of novel traits is there's a lot of challenges to studying this, particularly if you take a population biology approach, which is the way that most of my research is driven. So the first challenge is if we want to look at a novel trait, like this evolution of wings and bird example that I started with, The problem is we do not expect to have much of a signature of selection on these in current present day. So most of these traits have a deep evolutionary history. So wings did not evolve in species that are here today. They evolved in the ancestor of these species. So probably there may be some sort of tinkering with wing shape that we could detect as being under selection, but that broad trait itself is unlikely to be currently under selection. So an alternative approach we could take is we could compare genomes for a winged species versus a species that doesn't have wings. And so if we do this, we are going to pick up genomic regions that differ between these, but we're also going to pick up genomic regions that are responsible for a whole other suite of traits that differ between these organisms. And so teasing out which genes are responsible just for our trait of interest can be really, really challenging. So what we want is a system where essentially we have a newly evolved trait, but that's the only trait that differs between it and other species. Which is a pretty tough ask for a study system, except if you're the northern paper wasp. So this is Polistes fuscatus, and it is a super special species of paper wasp. So like other paper wasps, it is a primitively eusocial species. And so what this means is, unlike honeybees, in the spring, when it forms a nest, it can either do this by itself or it can form nests as part of these cooperative associations with other wasps. So these cooperative associations usually have a linear dominance hierarchy that they use to sort of organize reproduction. So eggs that are laid will develop into workers. Workers will help to sort of enlarge and build the nest. And then in the fall, reproductive individuals are going to start to emerge. So both our males and our future reproductive queens, which are called gines at this stage and foundresses at the other stage. So males and gines are going to die sort of at the end of the fall, and then the future queens are going to hibernate over the winter and sort of start the cycle all over again. So what makes Polistes fuscatus so unique is that all of the females have these individually distinctive markings on their face. So if you're not super familiar at looking at wasp faces, so we can focus on the sort of region in the front of the face here. So this is called the clypeus. So we can find individuals that have sort of all yellow clypeuses, sort of brown or pattern clypeuses, all black clypeuses. Here's another region that's kind of like an eyebrow-shaped pattern. We get some individuals that have like super thick eyebrows, some with really pencil-thin eyebrows, and some without this trait at all. And so the way that Fuscatus uses these unique markings on their face is to recognize and remember other females on the nest. And so this is actually the only known example that we have of individual recognition that occurs in insects. So here I'm showing you kind of what this would look like in nature. So this is a nest with a bunch of different Fuscatus on it. And if you look closely, you can see that they all have different patterns on their face. And essentially they function like name tags to other individuals. So allowing them to kind of keep track of who is pulling their weight, make sure everybody is sort of laying the appropriate number of eggs. So individual recognition is associated with the evolution of many cognitive traits. So wasps can remember more than 10 individuals, and they can remember them more than a week. And it's not that wasps in general are smarter than other paper wasps, it's that they're particularly specialized for the skill of learning faces. So they're not any better than other species at learning pattern differences, but they're way better at learning faces. Learning faces also seems to depend on color, so they're not as good if you ask them to learn black and white faces. And Fuscatus also shows improved visual acuity, suggesting that there's been some selection for the evolution of their ability to recognize these signals. Okay, so why is individual recognition beneficial? So Fuscatus has expanded their range much, much further north than other paper wasp species. So they're currently found into southern Ontario, kind of past where Toronto is. And there's a really interesting relationship between facial color pattern variation and latitude. And so this work was done by my former office mate and very talented postdoc who worked with me, James Tmolty. And so what he found is that as you go further north, we see an increase in this facial color pattern diversity. But if you go further south, and actually somewhere in the middle of maybe South Carolina, we reach a point where wasps shift, and actually the entire population has a sort of invariant red color face. And along with this sort of latitudinal gradient in facial color diversity, we find a difference in cooperative nesting ability, or cooperative nesting propensity. So those colorful wasps in upstate New York are going to be way more cooperative than their sort of less colorful southern neighbors. And so the benefit of individual recognition is thought to basically be to help wasps in these harsher winter environments that have sort of a shorter growing season, kind of get their nest going as quickly as possible. So the other thing that's nice about Fiscatus, going back to understanding individual recognition as a novel trait. So this trait has evolved recently. So it's not a trait that is present in these other species, but so you can see individuals from the same nest have sort of invariant faces. But these species in general tend to nest and mate and build, sort of fly around and live in the same habitats. So while there may be sort of other differences between the species, this individual recognition is going to be the main difference that we see. Okay, so returning to our expectations. So there could be two hypotheses going in. So we may see that individual recognition is a result of sort of weak selection spread across a lot of genes of small effect. Or we could see that this trait is a result of stronger selection spread across a fewer number of genes. So when we're talking about signatures of selection, so we can identify these using selective sweeps. So if this is not something you're familiar with, so here I'm showing a cartoon example of a population. So each of these dots is going to represent a variant. All of them are going to be neutral. And in this population, all of a sudden, we get the evolution of a beneficial novel mutation, this sort of star up at the top here. So at this point, if we scan across our genome, we see that there's sort of a consistent level of genetic diversity in our population. Makes it hard to detect selection. So this beneficial mutation will spread through our population, but along with a beneficial mutation, it's going to drag with it linked neutral mutations. And so this has an effect on genetic diversity. So now if we scan across our population, we see a decrease in our level of genetic diversity centered around this site. And then we can use this decrease in genetic diversity to detect a probability of selection there. So this can spread through our population, getting in what's called a hard selective sweep, so we can get this extreme reduction in genetic diversity. But over time, we can also see once hard selective sweeps have fixed, new mutations will start to develop on this background. In an older selective sweep, and we get kind of a reversal of this process. So we get sort of a smaller reduction in our genetic diversity. We can also get what's called a soft selective sweep, where our new mutation develops on multiple genetic backgrounds. And again, this has sort of a subtler effect on our ability to detect a reduction in genetic diversity. Okay, so with that in mind, the approach that we took for this study was to resequence multiple individuals, look for these signatures of selection, and then asked, are things that are under selection genes that we think would be related to our novel trait? So genes involved in learning, memory, and visual acuity. So we use this program called Sweep Finder 2, which outputs a statistic called a compositive likelihood ratio, or CLR score. That's essentially, so we thought that this might be associated with the strength of the selective sweeps, but we ran a couple of simulations to show that this is the case. And so essentially what I'm showing here is the value of this score across different strengths of selection coefficients. So essentially, all you need to take home for this is you get a higher CLR score when your selection is stronger. If we look at this over time, we see that the sort of the more in the past that selection took place, the lower our ability to detect the selection. And this is also true if we look instead of at a hard selective sweep at a soft selective sweep. So in general, there's also a lot of parameter space where I think it's a little bit hard to tease out, is this an old selective sweep? Is this a soft selective sweep? Is this a new selective sweep? But higher CLR scores are going to be associated with sweeps that are stronger and more recent. Okay, so what does this data look like? So we're going to look now across the entire genome of Plistes fuscatus. Each of these color bars is going to be a different chromosome, and we're getting that sort of CLR value on our y-axis. And it's hard to see kind of at the bottom, but that's actually our cutoff score for significance. And so we were really excited to see a bunch of really high peaks indicating signatures of selection. So in fact, we got 138 peaks that contained 183 genes. And something that was kind of unexpected going into the study, but it ended up being really advantageous, is paper wasps and other social insects have really high recombination rates. And this had this incredible effect of really narrowing down the regions that were under selection. And so in a lot of cases, we can be pretty precise that our signatures of selection are over sort of one big genic region, rather than other studies that have done this and have been over sort of tens of thousands of kb. So pretty cool. So also what was really nice is a lot of things that are under selection make sense for being involved in individual recognition. So we get a lot of genes here that seem to be involved in visual system development or long-term memory formation. So I'll kind of looking at this overall, so we concluded that there were about 39 good candidate genes for individual recognition within these windows. So I think it's a pretty clear support for more of an oligogenic selection regime. So yes. So something else we were concerned about, though, maybe there's something about life as a paper wasp that leads to selection on genes for learning and memory and vision that's going to be unrelated to this individual recognition trait. So we took advantage of these closely related species and essentially repeated this exact same analysis for those two other species I talked about, Metricus and dorsalis. And lo and behold, we find a very different pattern. So we still detect things that were under selection, but now our top windows are having things like odorant binding receptors, which may be important for species recognition or nestmate recognition, and a lot of things like transposable elements or retrotransposons. So we see sort of very different things. So we were also able to subsequently do this by looking at those southern populations I talked about that had invariant face colors. And essentially what we see in this case is there are some genes that are under selection in both populations. There are some genes that are under selection in each population. But those that are most important, that we'd annotated as being most important for this visual individual recognition, had much higher signatures of selection in our northern populations than our southern populations. So not involved in these sort of other species. Okay, so the other question that I asked about this trait, so are these alleles the result of new mutations in fuscatus? Are they segregating existing ancestral variation? And the way we did this was sort of two methods. So we did a bunch of population sampling for other species using whole genome sequence data, and then use this to classify the origin of alleles. So this is not going to work super well for really rare alleles, but essentially we can identify those within our data set that are only in fuscatus and that are either fixed or segregating. So to kind of complement this approach, we also used a machine learning approach to detect signatures of selection. It classifies regions in the genome as being under hard sweeps or soft sweeps or sort of neither. So if you throw this all together and we sort of look at those genes that I talked about as being candidates for individual recognition, what we find is that there are quite a number that are these de novo hard selective sweeps. So the de novo was going to represent that first method that I talk about looking at population sampling, and then the harder soft sweep is going to talk about that machine learning approach. So there are quite a number that sort of disagreed between the two methods, but at least in a number of cases, we can say that there's good evidence of new mutations in hard selective sweeps that are unique to Fiscatus. Okay, so lastly, about the timing of selection. So again, we can think sort of two alternative approaches to this. So maybe all of these genes were selected for in a pretty short, narrow window that could be related to our selection landscape, that there was some sort of vast shift, making it advantageous for all these alleles to fix. Or we can think of this as like a gradual process where they're sort of continually tinkering with this ability. So to address this, I use this program called STAR-TMRCA, where it essentially estimates the timing of origin of your alleles of interest. And so we did this for our candidate genes. So I think there are some caveats here. You know, these programs were not designed to work with haplodiploids. I think there's a lot of parameters that go into this that we're estimating without sort of real evidence behind what those parameters are. So I want to take this with a really large grain of salt, but I think there are a couple of things that we can conclude from it. So one, it does seem a little bit more of kind of a gradual saltatorial process. The other is our timing of it. So a lot of these are way before or way after the sort of last glacial maximum, which is a time that I would have thought maybe this trait could have evolved. So it seems to be very, very recent. If you want to be incredibly speculative, if you kind of squint, those genes that are older are more for discrimination, and those that are newer are more for cognitive ability. So maybe the ability to distinguish between them came first, and then the ability to remember those differences came second. But again, super speculative. Okay, so just to kind of sum up this, so we see a small number of genes, a mixture of alleles, but at least some of them seem to be coming from hard selective sweeps, and perhaps kind of a mixture of a gradual and punctuated process. Which I said here. Yeah, so we see recent song selection, loci not under selection, closely related species, and the discrete time periods. Great. Okay, so we're gonna completely switch gears now, get away from population genetics. Instead, now I want to talk about another source of variation. So how our abiotic environment can affect phenotypic diversity. So the trait that I'm going to talk about for this project is going to be body size. So differences in body size have important consequences for fitness. They can affect physiology. They can affect life history traits of organisms. There are, a huge number of studies sort of showing that body size varies with environment. So this is a classic example that I love. So if we compare cat body sizes in sort of low resources environments versus high resource environments, we see sort of healthy size versus major child cats. So body size also varies a lot geographically. So we see oftentimes many declines in body size across geographical regions. So I'm going to call these ecogeographical patterns. So there are a lot of hypotheses that have been proposed to explain ecogeographical patterns. So probably the hypotheses that you're going to be most familiar with is going to be Bergmann's rule. So we're going to be talking just about northern latitude studies in this. So latitude is going to be associated with decreasing temperature in all of these examples and in my data set. So Bergmann's rule suggests that body size gets larger as environments get colder. And so this was developed for endothermic organisms and it is essentially related to surface to volume ratio. So essentially having a smaller surface to volume ratio helps you stay warmer in cooler environments. So may not be super relevant for insects and other sort of ectothermal organisms. So we can get sort of similar patterns for other reasons. So one of these is the temperature size rule, which has to do with developmental plasticity and essentially suggests individuals in warmer environments reach maturity faster, but at a smaller body size than those in cooler environments. Or we could get it for something that's called the resistance hypothesis. So this suggests that essentially organisms that are larger are going to be more capable of surviving the winter when it's cold and when it's dry. And so that colder environments may favor the evolution of larger body sizes. We can also get predictions of sort of the opposite patterns. So one of these is due to the result of seasonality. So northern environments have sort of a shorter season to kind of get your offspring done and move on with your life. So we may see kind of the reverse of this temperature size rule. So a reduction in body size and a favoring of sort of fast development in colder climates. The other thing is that latitude co-varies with a number of other ecological factors, so including productivity, distribution of predators or other competitors. So we may see these ecogeographical patterns, and they may have nothing at all to do with temperature. We can also see sort of nonlinear patterns. So this is an example of what it would look like if we get a change in volcanism or the number of generations that are produced per year. So all sort of different possibilities. So what do ecogeographical patterns look like in insects? So there was a really nice meta-analysis done about 10 years ago that asked this question and looked across a number of orders of insects and essentially found that these patterns were highly idiosyncratic. So about a third of the time, they're showing a pattern that's consistent with a Bergmann's cline, a third of the time, a pattern that's consistent with a converse Bergmann's cline, and about a third of the time, a cline, like no cline at all. So this is a pretty broad group of organisms. So there could be quite a number of underlying mechanisms that are sort of driving these differences. And so what I really wanted to do is to look at these in a group of more closely related species. And so I thought, of course, paper wasps would be a great system to look at this in. So this is the distribution of paper wasps in the US. So each of these each species are going to be represented by a single color layer. So we see these sort of dense regions are going to be high species diversity. West Coast species are colored pink, East Coast species are colored blue, just to kind of help you keep track. So what's nice about this system is that we get multiple species with overlapping ranges that co-occur at different latitudes. And so kind of going into this, we could predict that if the same mechanism is driving these differences, that we would be able to pick it up across multiple species. The other thing that I really liked about using social insects to ask this question is social insects have these different casts that develop at different times of the year. And I thought that this might help tease out the sort of mechanisms that could be driving these differences. So for example, I talked about this resistance hypothesis about the need to be larger to help you survive the winter. But in the case of workers and male paper wasps, these are all going to die before the onset of winter. So this could be not a selective factor for them. So it seemed like a cool way to kind of tease these out. The other thing that's nice about paper wasps, maybe not great for paper wasp ecology, but nice for the study design, is we have an invasive species, the European paper wasp, that appeared in the 1970s. And so if it were to show variation in latitude, I think this would strongly suggest that there is some degree of phenotypic plasticity that might be driving these changes. So the way that I addressed this issue was using a method that I developed looking at high throughput measurements of body size from natural history collections. So essentially went through and took photographs of these whole boxes of pinned specimens and then did a multivariate measurement of body size. So, natural history collections are a little bit random about where species are collected and when and kind of the distribution of those. And to get around that randomness, I just threw a bunch of data at this problem. So I was able to get measurements for about 15,000 different specimens. Okay, so what does this look like? So I'm going to show you this for one species, Polistes annularis. So we get our measurement of body size versus latitude, and pretty strong result of a converse Bergmann's cline. What does it look like for other species? looks something like this. So instead of getting converse Bergmann's climbs for everybody, again, I'm getting sort of an idiosyncratic pattern. So seeing really strong ecogeographical patterns for most of these are significant, but the direction of the climb varies a lot. I also parsed this data out looking at casts. I looked at elevation instead of latitude. I looked at bioclimatic variables instead of latitude as well, and essentially get kind of the same pattern. And so for reference, this is that invasive species that I talked about. So it is showing a converse Bergmann's climb. So I thought, maybe this difference has to be a phylogenetic difference, that there was some sort of transition between species that had a Bergmann's climb or a converse Bergmann's climb. And the answer to that is also no. So kind of mapping this onto a phylogeny, both our body size and the direction of this climb, we find no signal in a phylogenetic signal in Bergmann's climbs. There is kind of an interesting finding where body size measurements are going to be more divergent than expected under random drift, which I think is kind of intriguing. And kind of leads me to a direction that I'd like to take with this in the future. So what I'm showing you here now is all of this data just for those East Coast species. So the lines have been truncated just to show the sort of species distribution. The direction, this is the slope of the regression line between body size and latitude. And it's separated out for our full data set for spring, which are going to be those foundresses, for summer, which are going to be workers, and for the males in our data set. So the dotted lines are those that did not quite reach significance. The solid lines are those that are significant. And so, again, I think if you kind of stand back and squint, at least what this suggests to me is a pattern that looks a lot like trait overdispersion, or sometimes called constant size ratios, which essentially says that our species within the same guild are evenly distributing themselves through phenotypic space. And this pattern is one that would be consistent with divergent character displacement. So this is something that I'm hoping to follow up with more experimentally in my lab. But I think that there's at least something to suggest that that body size of wasps has to do with sort of the composition of the species in the environment. So just what did I show you here? So paper wasps in North America have very strong ecogeographical patterns in their body size, but these patterns are really idiosyncratic. So they're unrelated to phylogeny, still have no idea what these mechanisms are. We get similar patterns in both workers and reproductive castes. And body size might be related to the presence of other species. Okay, so the last project that I'm going to tell you about, we're going to kind of zoom out even further and look now across all paper wasps and ask about biotic interactions. So I started earlier in the talk with this slide. So what I'm showing you here, each of these images is a female of a different species of paper wasp. So if nothing else you take away from this talk, I hope that in the future, when you're looking for an example of a highly colored, diverse species, instead of something lame like birds, you can think of wasps instead, the flying animal of choice. So they show both a wide variation of color pattern variation in their bodies and in their faces. So for faces, I think at least there's a really strong case to be made that these are for signaling to other wasp species. So they're going to be really small. So this is not something that their predators are going to notice from any distance away. Wasps also spend a lot of time kind of very close to each other, kind of interacting with each other. And we get multiple species that co-occur in the same area, offering opportunity for species interactions. So there are a multitude of different ways that paper wasps could be using these facial color patterns, interspecific interactions, telling differences between males and females. They could be using them to mediate cooperation and conflict, like I talked about in Felistes fuscatus. But they could also be using these in interspecific interactions in species recognition. And what I think is cool about paper wasps as well is they need to do species recognition in mate choice contexts, like normal organisms do, identifying the right mate. But paper wasps, also because they have the cooperative nesting associations, are also going to need to do species recognition at that point as well. In the interest of time, I'm going to ignore all of my intraspecific and concentrate on the interspecific variation. But I have a bunch of projects I'd love to do with this in the future. So again, the data set that I'm going to talk about is based on museum collections. So in this case, I have about 16,000 photographs from about 131 species. So there are somewhere in the neighborhood of 200 to 300 species of paper wasp, depending on if you're a lumper or a splitter. I'm a taxonomic lumper, so I'm going to ignore all of my subspecies and kind of group them together. So have somewhere in the neighborhood of 1 to 30 specimens per species, with on average, about 12. And just as a little bit of a shorthand that we can use for species that I don't have phylogenetic information for, paper wasps can be split into these 4 kind of regionally restricted subgenera. So there's a New World and sort of 2 Old World, or 3 Old World genera as well. Okay, so when you want to compare facial color pattern diversity across this wide range of species, we want to come up with a way to describe this color pattern diversity. And museum specimens present quite a number of challenges. So specimens are often pinned in slightly different directions. We can see that they're just like antennae all over the place. And essentially collection managers aren't really thrilled about you decapitating all of their specimens, so you can take really nice standardized pictures. So to get around this, what we ended up taking is what we're going to call a guidebook style approach. And so we picked kind of a representative female from our specimens of each species. For reasons that I don't have time to talk about, we're going to restrict our color palettes to yellow, red, brown, or black. And then essentially, we translated the color pattern on the specimen to a standardized WAPS color face. So a lot of this was done with the help of a really talented undergraduate student, Lily Woodward. And most of the subsequent analysis we've done using this package called Patternize, which is really useful. So one of the benefits of using this sort of standardized face is that it removes any sort of variation in size and allows us just to focus on facial color patterns. And also by using the standardized face, we essentially have a color value at each pixel for each species. And so we can take all of these color values for pixels and put them into a principal component analysis. So that essentially looks something like this. So this is our principal component analysis of paper wasp facial color diversity. So the first 2 principal components are actually representing quite a bit of variation, so nearly 70% of our variation. I have these sort of cartoon images below so you can see what that first and second principal component analysis are actually representing in sort of facial color pattern space. And some of the way that this worked out, so all of our tips are going to be solid colored faces, and there are quite a number of species that are sort of stacked on top of each other there. So solid colored face was the most common face facial color pattern. So the couple of really interesting insights that jump out from this. So first, wasps aren't using all of the potential facial color space. There's quite a bit of space that's not occupied. So we get a region here where we don't find very many species. There are also color patterns that we don't see. So we see species that are red with red jaws and black with red jaws, but we never saw a species that's yellow with red jaws. So I think representing that there may be some sort of developmental or preference constraint on wasp facial color diversity. We also see the kind of repeated evolution of patterns. So in this case, this yellow marking on Eclypius with sort of yellow markings on the inner eyes has evolved twice. And the colored arrows here and that I'm going to be using on other slides are going to represent those different subgenera that I talked about. Yeah, so taking more of a global look about this. So are species that are falling closer to each other in this facial color pattern space, are they going to be more closely related to each other, or are they going to be farther away? So is each of these going to be a subgenera? Question mark. And the answer to that is kind of no. So I'm going to surround the facial space occupied by each subgenera in these different colors. And so essentially we can see that the subgenera are using almost the entirety of face space that happens. So it's an interesting example of parallelism. And then we get an even more intriguing example of parallelism here, where we get species that occur in completely different regions seeming to recapitulate almost the same facial color patterns. So on the top, we have a species from China and a species from the US. On the bottom, we have a species from the Philippines and a species from Central and South America that essentially are in different subgenera, but have the same facial color pattern variation. So again, I think suggesting that there are some patterns that are just really exciting to have, or some sort of developmental constraint, or both, on what patterns we're getting. Okay, so instead of looking at subgenera, I want to zoom in a little bit, or look across all species. I'm going to zoom in and look just at this one specific subgenera that we have phylogenetic information for these species. So off to the side, I'm showing all of the species that I'm going to show here. Just going to look at their sort of differences in facial color patterns. And I have them mapped here on that principal component space. So when we just look at our distributions of species, we see, some kind of are closer together, some are sort of further apart. But now I'm going to map on our phylogeny on top of this, and it looks something like this. And so now what I think we can see, this is something I'd like to do a little bit more quantitatively. I'm still kind of working my way through some of this data, but at least for visualization purposes, we see sister species are quite far apart now on phenotypic space. So our marginalis is all the way over here to Big Loomis, which is up here. And we get, Riparius and Nymphus are next to each other here, but they're not very close to each other on the phylogeny. So I think this, at least to me, suggests the potential that wasps are selecting to be distinct in their phenotypic space. And this is something that I'd like to do quite a bit more of in the future. So there's one other cool thing that we can do with this data set. So because we have all of these values at different pixels, we can take our value for each of our principal component analysis and use them to do ancestral state reconstruction. So this is, again, that same sort of clade that I was showing. And so we use the first 26 principal components because this seemed to give us kind of the best results, and constructed what we would predict our ancestral face would look like. And it looks something like this. So those regions with solid color had really high support for that color. Those regions that are kind of in between, it could go either way for whether or not it was black or yellow at these. But I think gives a pretty convincing result, so that the ancestor for this clade seemed to be black and yellow, seemed to have some sort of pattern on the front of its clypeus, and seemed to have some sort of eyebrow. You can do this, again, across the entire phylogeny. I think the data gets a little bit sketchier as you kind of zoom out. Or more speculative, maybe I should say. But I do think that from this, what you can take is that the ancestor in the base of this clade seems to be pretty strongly a wasp that had more than one color. And so that facial color patterns are most likely ancestral for paper wasps. Yeah, so paper wasps have highly diverse facial color patterns. These components have evolved independently multiple times. There is some evidence in at least some clades that there's evolution on the ability to be distinct in your facial color patterns. We get an ancestral face that looks like this. And this is a really exciting data set that I'm looking to explore more. I also have a whole suite of body color patterns too, that we'll get into this at some point. So yeah, so I've told you a number of stories today about paper wasps and the origins of diversity. So I've shown the evolution of individual recognition, which is a novel trait in Polistes fiscatus. We looked at abiotic environments and how body size seems to vary with the environment, but the sort of mechanism behind that seems to change from species to species and is yet unclear. And then we talked about how paper wasps facial color pattern diversity may be responding to selection from the presence of other species. So with that, I have a lot of people to thank. In particular, I want to thank my postdoctoral advisor, Michael Sheehan, and my office mate, James Tumulty, who helped me, was a key component to a lot of these analyses, members of the laser lab at Cornell, and a bunch of the natural history collections where I got this data from. And I'll take any questions.
All right, I see a few hands. I'll start here.
Really, that's not. I'm not even sure where to start. But let's go with the body color thing. So in my experience, although I haven't looked at all those species, the colors you have on your face are probably the colors you got on your body. And then when you think about body coloration, you think, I think first organ coloration, perhaps.
Mhm.
So are you planning to look at that?
Yeah, so I would say that I would agree with you that most species seem to have the same color on their body as their face, but it's not been something that's looked at, and I can think of exceptions to that rule. And so I'd like to dig a little bit deeper on this idea of developmental constraints to see, which facial color patterns and which body color patterns, and if there are, how those are associated with each other. So something that I'd like to explore more in the future.
And if you could just repeat the question.
Yes, thank you.
Thank you so much. It was a fantastic talk. I have two questions, and they both revolve around your approaches with machine learning. So the first one has to do with your identifications of selective sweeps. So I was curious, do you have a good idea of sort of the crown divergence times for your group, and how did that inform your usage of the animal method for identifying selective sweeps?
Yeah, so the question was about the methods that I used for identifying selective sweeps. And so working with a non-model species, and especially a species in Hymenopteron where we have haplodiploids, there's really not much that's known about crown ages or sort of model parameters. And so I would say a lot of it was kind of guesstimate, which was our approach of throwing a bunch of analyses at it.
The second question, how much of this, I mean, so many specimens, how much of it was made possible because you could use high throughput methods to image all of these specimens you were looking at it? Did you use machine learning methods to sort of do that work? Or?
No, so this was about machine learning and museum specimens. So I didn't use any sort of machine learning for that. That kind of, based on my experiences and others, I felt like we would spend more time kind of fiddling with the data. There are other analyses looking at color pattern diversity that are coming out of the Sheehan lab that might use some of those approaches in the future. So stay tuned there.
Yeah, I really enjoyed the talk.
Thank you.
I had a question about the facial recognition.
Yeah.
Do wasps use facial recognition during mating? Because I was just thinking about, maybe it's a way to increase genetic diversity and avoid inbreeding.
Yeah, so the question was about facial recognition during mating. And so I didn't have, I ended up axing this for my talk for time, but actually most species of paper wasps, including fiscatus, males have yellow faces, so look completely different. So males aren't showing any of this facial color pattern diversity at all. This is a property of females. There are studies in other species that have noticed differences in other parts of the coloration, other parts of the body that might be associated with mate choice. So that invasive species that I talked about, the size of some of the black spots seem to indicate higher quality males for mating with, but in general, males don't do individual recognition. So.
We have a couple of questions on YouTube from John Birmingham Jr. And the first, which was submitted during the first portion of your talk, asks, does olfaction have any role in individual recognition? Yeah, so does olfaction have a role in individual recognition? So olfaction is really important in social insects and certainly has a role in recognition of species and also nest mate recognition. It has not been specifically tested for individual recognition, but the studies that were done originally to show individual recognition were done using images. So wasps do not need olfaction to recognize, but that doesn't mean that olfaction may not play a role. Thank you. And then the second question was, have you looked at higher principal components, i.e. third, fourth, et cetera, to see if they help with deeper biology? Yeah, so we have looked at higher principal components. There's a lot I'm still kind of digging into that analysis. If you get much higher principal components, they start to be things that are represented only in a single species. So the 26 principal components does a pretty good job of summarizing facial color variation.
Great talk. If I understood correctly, in the first part of your talk, you mentioned an interest-specific latitudinal gradient in Polyspes fuscatus in facial color variation, where there's a benefit to having variable facial color in the north, but that variability goes away in the south. I'm curious why that variation goes away in the south. Is there a cost associated with it?
Yeah, so the question is about this transition in facial color pattern variation between the north and the south. So yeah, I think that this is something that needs more study. I mean, there's definitely a transition, a cost to reproduction, right, when you're cooperative. So when you're having to share your reproduction with other females, you're producing fewer offspring than when you raise a nest yourself. And so some of this may have to do with the fact that there's a transition, kind of an inflection point where most nests are going to be successful when you rear them by yourself versus most nests you're going to need help to get those nests going and to raise them to the next generation. But kind of other than that, we're not particularly sure.
I wanted to ask a question. It's kind of a statistical question. It's about Bergman's rule, a very general one. It seemed like about a third followed it, and a third followed the opposite, and a third didn't show a pattern. And I'm just imagining a null model where different species, like the average species, never patterned, but kind of by chance some did and some didn't. And how would you differentiate, there's real deviation, or this one really follows, this one really doesn't, versus have a null approach?
Yeah, so whether or not you should use a null model to address the Bergman's rule patterns, I'd have to think about that more. I guess I would be surprised that you would get a significant association for every species if this was kind of the result of some sort of random chance. But I think that really experimental approaches are kind of needed to tease out the mechanism. That's sort of what I want to do, is raise wasps in warmer and colder environments and see if they really do get bigger. But yeah, if you have ideas about statistics that I should be employing, I'd be happy to hear that.
I know sometimes if you get a whole lot of data, you can have very low R squared and the slope's very close to 0, but you have enough data that P is less than 0.5. And I was just thinking about these things when I saw it. But
yeah.
Great, Sarah.
Great, thanks. Thinking again about that Bergman's rule portion of your talk and that invasive species. And so I think you showed a pretty strong inverse Bergman's pattern with that species. And am I right in assuming was that tested in the invasive range?
Yeah, so this is again sort of repeat your question. So this has to do with the Bergman's rule in the invasive species. So a lot of that has to do with kind of the vagaries of museum data. So this species is actually found now throughout the entire US, but I was only able to get specimens that people collected it from kind of the New York, Canada, upstate area. But it does show changes there. So might be doing different things in different parts of its range.
Okay, yeah, so my mind went to the possible comparisons between the native and invasive ranges, if there's different numbers of co-occurring species, and that might be able to get at some of your hypotheses about the number of like co-occurring species, trait over dispersion.
Yeah, so I have played with some of the trait over dispersion data with the museum collections and essentially just found that I didn't have enough power with the sample that I have. And so it's really something I think we're gonna have to do experiments on in the future, so.
Hi there, yeah, great follow-up. Thanks very much. I was just few interrelated questions. So in the northern latitudes, the ones that do have the kind of interspecific variation basis, the lower wetter are assuming just kind of interrelated individually.
Yeah, so all paper wasp species in northern latitudes are going to overwinter for the most part. So this is not unique to Fiscatus. Yeah.
So the subsequent, during the spring, when they're finding their nests and they kind of come together, do you find that they're kind of forming nests within more so than the population magic? And if that's the case, then could kind of the facial recognition be kind of like during the kind of during their early life when they're being raised as a nest?
Yeah, so the question is about, are Fiscatus forming nests more often with kin? And I'd say that if you asked this question to somebody 20 years ago, they would have said yes, that most often wasps in general and Fiscatus are nesting with their sisters. I would say that I've done a lot of sequencing of wasps now, and we often find individuals on the same nest that are not related to each other or that are more like distant cousins. I have a pet hypothesis that this is different in Fiscatus than in other species. So essentially that they're forming cooperative associations with individuals they're not related to. But we haven't done enough population sampling of other species to really support my pet hypothesis yet. So stay tuned.
Okay, it is now 5 o'clock. So if you have any more questions, please join us across the hall for refreshments.